Open Stax Psychology Chapter 5 continued. Section 5.3 Visions. The visual system constructs a mental representation of the world around us. This contributes to our ability to successfully navigate through physical spaces and interact with important individuals and objects in our environments. This section will provide an overview of the basic anatomy and functions of the visual system. In addition, we will explore our ability to perceive color and depth. Anatomy of the visual system. The eye is the major sensory organ involved in vision. Light waves are transmitted across the cornea and center the eye through the pupil. The cornea is the transparent covering over the eye. It serves as the barrier between the inner eye and the outside world, and it is involved in focusing light waves that enter the eye. The pupil is a small opening in the eye through which light passes, and the size of the pupil can change as a function of light levels as well as emotional arousal. When light levels are low, the pupil will become dilated or expanded to allow more light to enter the eye. When light levels are high, the pupil will constrict or become smaller to reduce the amount of light that enters the eye. The pupil's size is controlled by muscles that are connected to the iris, which is the colored portion of the eye. After passing through the pupil, light crosses the lens, a curved, transparent structure that serves to provide additional focus. The lens is attached to muscles that can change its shape to aid in focusing light that is reflected from near or far objects. In a normal-sighted individual, the lens will focus images perfectly on a small indentation in the back of the eye known as the fovea, which is part of the retina, the light-sensitive lining of the eye. The fovea contains densely packed specialized photoreceptor cells. These photoreceptor cells, known as cones, are light-detecting cells. The cones are specialized types of photoreceptors that work best in bright light conditions. Cones are very sensitive to acute details and provide tremendous spatial resolution. They also are directly involved in our ability to perceive color. While cones are concentrated in the fovea, where images tend to be focused, rods, another type of photoreceptors, are located throughout the remainder of the retina. Rods are specialized photoreceptors that work well in low light conditions, and while they lack the spatial resolution and color functioning of the cones, they are involved in our vision in dimly lit environments, as well as in our perception of movements or the periphery of our vision field. We have all experienced the different sensitivities of rods and cones while making the transitions from brightly lit environments to a dimly lit environment. Imagine going to see a blockbuster movie on a clear summer day. As you walk from brightly lit lobby into the dark theater, you notice that you immediately have difficulty seeing much of anything. After a few minutes, you begin to adjust to the darkness and can see the interior of the theater. In the bright environments, your vision was dominated primarily by cone activity. As you move to the dark environments, rod activity dominates, but there is a delay in transitioning between the phases. If your rods do not transform light into nerve impulses as easily and efficiently as they should, you will have difficulty seeing in dim light, a condition known as night blindness. Rods and cones are connected to retinal ganglion cells. Axons from the retinal ganglion cells converge and exit through the back of the eye from the optic nerve. The optic nerve carries visual information from the retina to the brain. There is a point in the visual field called the blind spot. Even when light from a small object is focused on the blind spot, we do not see it. We are not consciously aware of our blind spots for two reasons. First, each eye gets a slightly different view of the visual field. Therefore, the blind spots do not overlap. Second, our visual system fills in the blind spots so that although we cannot respond to the visual information that occurs in the portion of the visual field, we are also not aware that information is missing. The optic nerve from each eye merges just below the brain at a point called the optic chiasm. As figure 5.12 shows, the optic chiasm is an X-shaped structure that sits just below the cerebral cortex at the front of the brain. At that point of the optic chiasm, information from the right visual field, which comes from both eyes, is sent to the left side of the brain, and information from the left visual field is sent to the right side of the brain. Once inside the brain, visual information is sent via a number of structures to the occipital lobe at the back of the brain for processing. Visual information might be processed in parallel pathways, which can generally be described as the what pathways and the where, how pathways. The what pathways is involved in objects recognition and identification, while the where and how pathways is involved with location in space and how one might interact with a particular visual stimulus. For example, when you see a ball rolling down the street, the what pathway identifies what the object is, and the where how pathway identifies its location or movement in space. Color and depth perception. We do not see the world in black and white, neither do we see it as two-dimensional or flat. Let's look at how color vision works and how we perceive three-dimension. Color vision. Normal-sighted individuals have 3 different types of cones that mediate color vision. Each of these cones types is maximally sensitive to a slightly different wavelength of light. According to the trichromatic theory of color vision shown in figure 5.13, all color in the spectrum can be produced by combining red, green, and blue. The 3 types of cones are each receptive to one of the colors. The trichromatic theory of color vision is not the only theory. Another major theory of color vision is known as the opponent's process theory. According to this theory, color is coded in opponent's pairs: black, white, yellow, blue, and green, red. The basic idea is that some cells of the visual system are excited by one of the opponent colors and inhibited by the other. I think it does. Yeah. So a cell that was excited by wavelengths associated with green would be inhibited by wavelengths associated with red, and vice versa. One of the implications of opponent processing is that we do not experience greenish reds or yellowish blues as colors. Another implication is that this leads to the experience of negative afterimages. An afterimage describes the continuation of a visual sensation after removal of the stimulus. For example, when you stare briefly at the sun and then look away from it, you may still perceive a spot of light, although the stimulus has been removed. When color is involved in the stimulus, the color pairings identified in the opponent's process theory lead to a negative afterimage. You can test the concept using the flag in figure 5.14. But these 2 theories, the trichromatic theory of color vision and the opponent's process theory, are not mutually exclusive. Research has shown that they just apply to different levels of the nervous system. For visual processing on the retina, trichromatic theory applies. The cones are responsive to 3 different wavelengths that represent red, blue, and green. But once the signal moves past the retina on its way to the brain, the cells respond in a way consistent with the opponent's process theory. Depth perception. Our ability to perceive spatial relationship in 3-dimensional spaces is known as depth perception. With depth perception, we can describe things as being in front, behind, above, below, or to the side of things. Our world is 3-dimensional, so it makes sense that our mental representation of the world has 3-dimensional properties. We use a variety of cues in a visual scene to establish our sense of depth. Some of these are binocular cues, which means that they rely on the use of both eyes. One example of a binuclear depth cue is binuclear disparity, the slightly different views of the world that each of our eyes receive. To experience this slightly different view, do this simple exercise. Extend your arms fully and extend one of your fingers and focus on that finger. Now close your left eye without moving your head. Then open your left eye and close your right eye without moving your head. You will notice that your finger seems to shift as you alternate between the 2 eyes because of the slightly different view each eye has to your finger. A 3D movie works on the same principle. The special glasses you wear allow the 2 slightly different images projected onto the screen to be seen separately by your left and your right eye. As your brain processes these images, you have the illusion that leaping animals or running persons is coming up towards you. Although we rely on binocular cues to experience depth in our 3D world, we can also perceive depth in 2D arrays. Think about all the paintings and photographs you have seen. Generally, you pick up on depth in these images even though the visual stimulus is 2D. When we do this, we are relying on a number of monocular cues, or cues that require only one eye. If you think you can't see depth with one eye, note that you don't bump into things when using only one eye while walking, and in fact, we have more monocular cues than binocular cues. An example of monocular cues would be what is now known as linear perspective. Linear perspective refers to the fact that we perceive depth when we see two parallel lines that seem to converge in an image. Some other monocular depth cues are interpositions, the partial overlap of objects, and the relative size and closeness of images to the horizon.